Hello everyone, my name is Daniel and I am a TA for the BIO107 virtual course. Today I'll be covering the concept of photosynthesis and how it may apply to the virtual labs you are to complete for this week. Well, first it's important to understand that CO2 can be fixed by plants and some bacteria. This process is called photosynthesis, which is the fixing of carbon from the environment to produce a usable form of energy. In most cases, this form of energy is going to be glucose. Almost all plants are characterized as photosynthetic autotrophs. This means that they can produce their own food through photosynthesis, and some organisms can use other means of creating food, such as chemosynthesis. It is important to understand that photosynthesis is primarily a light-driven process, where energy that is transported by light is used to convert atmospheric CO2 into sugar. The plant you see below is called Elodia, and it is a freshwater plant which is fast growing. It has very large vesicles, so we use them in regular lab courses as well as the online virtual course. So this Elodia plant down here on the left. And what you're seeing on the right here is the general reaction for photosynthesis, where we have carbon dioxide going in, water going in, and light energy going into the system. From this system, you are, in theory, supposed to produce glucose and oxygen. So to understand photosynthesis, it is first important to understand light. So visible and non-visible light is characterized as electromagnetic radiation. This radiation transports energy throughout the universe in the form of photons along a wave. And below is a diagram of a electromagnetic spectrum. So a spectrum shows you a spectrum of various lights. It shows you all the wavelengths of light rays and their respective energies. So a combination of all of these wavelengths creates a visible white light. When you see a rainbow, so your Roy G. Biv classic colors, what's happening there is your white light is being split into its various wavelengths. So you're seeing the blues, you're seeing the greens, you're seeing the yellows and the reds and everything in between. Now this is caused by molecules of water splitting the light that's coming through the atmosphere. Now, one important property of light and its wavelengths is that the energy of a wave is inversely proportional to wavelength. So as you have longer wavelengths, such as radio waves that we listen to our radios play in, those do not have high energy. Therefore, they are relatively safe for us to use. Now, on the other side of that spectrum, we have gamma rays. Gamma rays are extremely short wavelength and very, very high energy wavelengths. So in the case of the Hulk, it did not work out well for him. And it would not work out well for us either. So you want to avoid these short wavelength, high energy rays as much as possible. But for our purposes, they are actually useful because they allow plants to grow significantly faster. Now we'll discuss why that is. So now that you have an idea of what light is and how it's actually characterized, let's see how it might actually be absorbed or used. So the plant we're looking at today in all of our experiments is going to be Elodia. So Elodia, as you can see, contains many cells. This is its cellulose cell wall. And what you're seeing here within the green capsules here are chloroplast organelles. So chloroplasts are floating around, they move freely within the cell wall, and within them are embedded photopigments. So these photopigments, such as chlorophyll A, B, and various other carotenoids, are responsible for directly absorbing any potential light energy that comes into contact with the plant cell surface. So they are housed within what is called these thylakoid stacks. So as you can see in the picture to the right, this is a chloroplast cross-section revealing these stacks of green, essentially going to be chlorophyll housing units. So these are going to be our energy reservoirs. So one might ask how these were actually characterized. How do we know they're there? Well, one way we figured this out was using paper chromatography. So about 200 plus years ago, this technology was discovered, and to this day we still use it in some of our chem labs. So in this process, we apply essentially blended plant material onto a onto a chromatography paper, and then we dip this chromatography paper into chromatography solvent. The solvent is generally going to be an ether or some sort of alcohol that can dissolve and solubilize the pigments. So once this paper comes into contact with this solvent, as you see down here to the left, once it comes into contact, this solvent will start to move upwards into the paper until it reaches a designated mark position. Once it starts moving up, all of the material that has solubilized into the solution will begin to move up with it. So pigments which are less soluble will stop sooner, and pigments which are more soluble will stop later on. This causes essentially a separation. And once you have these separated, you can then physically cut them out and put them under a scrutiny of experiments to determine what their activity actually is. So once they did determine what their activity was, they assembled what is called an absorption spectrum, or an absorbance spectrum. And what you're seeing here are chlorophyll A, chlorophyll B, and your carotenoids. Now your carotenoids are various other pigments, but they're essentially the red, greenish guys. So what you're looking at is the absorbance wavelengths for these various photopigments. So chlorophyll A will absorb all the light within this larger range, as well as this end between about 650 to 700. Chlorophyll B will absorb from anywhere from, looks like 400 till maybe 470, and then again on the other end. And the carotenoids again will absorb at their designated location. The important aspect is to see that there is no absorption at the green wavelength. And this is why we see green colors on our leaves, because no absorption means reflection. So if there is no absorbance, we will see green. Whatever you are seeing is what is not being absorbed, and that's an important concept to understand. So now that we have a good understanding of what goes into photosynthesis, what comes out of photosynthesis, what kind of light is used in photosynthesis and what kind of light is not used in photosynthesis, I think you are now prepared to do the virtual labs. So go ahead and get ready to do the photosynthetic pigments experiment, the white light and green light experiments. But before we actually dip into that, we're going to go over what a volumeter is, because most of you have probably never used one of these things. So a volumeter is just what it sounds like. It's a meter used to calculate volume within a cylinder or a tube. In this case, we are using test tubes with a volumeter attached to a stopper. So once you set up your experiment, as I have in my virtual lab, you will see these numbers start to change over time. Now, your goal for all the labs using these volumeters is to determine what is coming out of this chamber and what is potentially going into this chamber. So what is being produced, what is being used, that is your responsibility for these virtual labs, is to come to a conclusion as to what is happening given the information we went over. Once you have completed those labs, you're going to come back to this video and complete the rest of the photosynthetic lecture. So now that you've gone over all the light reactions as well as paper chromatography, I'd like to go into a little bit more in depth of what the actual reaction is for photosynthesis, specifically what is actually happening within the chloroplast, what's happening within the energy factories, within these energy reservoirs we're calling the chloroplast. So what's happening within the chloroplast is a set of reactions called the dark reactions and the light reactions. They work in parallel together to produce this system, which in the end produces sugars. Now, again, the first half of the reaction, of the basic reaction, is water, carbon dioxide, and light. So they do not go in any specific order. They are happening at the same exact time. So let's just begin with the left side. So the left side is the light reactions. In this case, we have water coming into the system and light. Light will excite your photopigments that are absorbing that light energy in the form of photons. And through this excitement, they will create ATP, NADPH, and release oxygen as a waste product. I say waste product because that's what it is in this reaction. It is a great product, but it is a waste product. Now, the Calvin cycle is responsible for directly converting that CO2 that is going into the system into usable sugars, while also reducing the ATP and NADPH from your light reactions. Now, it needs to reduce these ATP and NADPHs because that is where the chemical energy is being stored. The chemical energy is being stored within the ATP and within the NADPH. So when these two molecules go into the Calvin cycle with carbon dioxide, they will begin to form sugar. Now, why do we even want sugar? Well, for many reasons. Sugar is involved in many energy processes, so cellular respiration, creating cellulose for structure, creating starch for energy storage, and essentially all other organic compounds are going to be derivative of sugars. And now that we've taken a more in-depth look into photosynthesis and we know what comes in and out of the system, one might ask, does photosynthesis affect pH? Now, why might somebody ask this? Well, because of global warming. So global warming has caused the ocean acidity to increase significantly. This is due to this chemical reaction that's occurring between water and carbon dioxide. It converts it into carbonic acid, which then increases the acidity of the ocean. So one might speculate that photosynthetic organisms have a role in reducing this effect of carbonic acid. Well, how might one test for this? Do photosynthetic organisms, water organisms, remove acidic carbon compounds such as carbonic acid from their environment? And if so, what might that rate be? So we can test this using phenol red. And you will test this using phenol red in your virtual lab. You're going to assemble a solution with a plant in it, and you're going to see what color changes over time occur. Now, if your phenol red, which is a pH indicator, turns yellow, then it is acidic. If it turns red, then it is basic. So you want to look for those changes that occur. And it's important to understand and remember that if you introduce carbon dioxide to water, it will become more acidic. So in the virtual lab, you are asked to breathe into the water. That is your introduction of carbon dioxide into the water. With that, I will be ending this virtual lecture. I appreciate you taking the time to listen, and we'll see you next week.